listen. She's going to ask it. Ready?
My question was, does creatine phosphate give something ATP to make more ATP?
Yeah, so creatine phosphate, remember, was the second place where the cell goes for energy, because phosphate groups are what are needed to make more energy. So once you've burned the stored energy, adenosine triphosphate, then if you can very quickly add phosphate groups to ADP, you have more energy. Creatine phosphate is a molecule that is stored in the muscle as extra phosphate groups. And as soon as you burn the phosphates that are stored, the creatine will give its phosphate groups to ADP with the use of an enzyme called creatine phosphokinase. And so you will very quickly make additional energy. So you use stored ATP first, then you use up the phosphate groups from creatine phosphate second, and then third, you'll have to make energy. And you're going to do that hopefully using aerobic respiration. That's a good question. All right. Is that good? Is that all of it? Okay. Okay, so the nervous system is the last piece of exam 3. So we've already done a lot of nervous system stuff, but I want to give you some outline features of the nervous system and teach you a little more anatomy about how the nervous system is constructed. So this picture tells you that the nervous system is made up of several pieces and parts. You can see at the top of the picture here the brain and the spinal cord. We say that the spinal cord can be we say that the nervous system can be described anatomically and physiologically. If I describe the nervous system anatomically, I say that it has 2 major divisions, a central nervous system and a peripheral nervous system. The central nervous system is the brain and spinal cord. The peripheral nervous system are the cranial and spinal nerves. So we divide the nervous system up structurally into the central and peripheral components. There are 12 pairs of cranial nerves and 31 pairs of spinal nerves. I would like for you, before we finish talking about nervous system stuff, we're going to do this more in the next chapter, so this won't be on this exam. I'd like for you to learn the names of the cranial nerves and something about each one of them. So we'll do that later. You have already been introduced to this concept that there are 31 pairs of spinal nerves, because I taught you that this is how you get information from the brain to the skeletal muscles. So in our tracking of information through the system, we detailed some of this pathway stuff using the spinal nerves, cervical, thoracic, lumbar, and sacral nerves. Okay, so I can talk about the nervous system as being divided into I can talk about the nervous system in anatomical divisions, the central nervous system and the peripheral nervous system. Or I can talk about the nervous system in functional divisions, physiological divisions. And those divisions I would call the somatic and the autonomic. Now you've heard these words already, somatic and autonomic. But I'm going to give you a little bit more today about what I mean by somatic and autonomic. In the first test section of this class, I defined them for you. I said, when I say somatic nervous system, you should think skeletal muscle contraction. When I say autonomic nervous system, you should think cardiac muscle, smooth muscle, and gland function. So we even went so far as to say the autonomic nervous system is parasympathetic and sympathetic. Okay, we need to do a little better than that. Okay, now this picture is intended to drive home the image of the connections made between the peripheral and central nervous systems. So these are fundamentals that I'm going to use to teach you some of the basic anatomy of these systems some of which you already know. Okay, so the first part of this picture you can see is showing a connection here between receptors and the spinal cord. So what you learned when I first started talking about nervous system stuff, we talked about this idea that you have information coming to and going away from the nervous system. All right. So image in your head of a neuron here that is bringing sensory information to the central nervous system. This information out here would be part of the peripheral nervous system, bringing information to the central nervous system. This is sensory information. And this is going to be the topic of a slide coming in just a few minutes, where I show you the sensory neurons and their primary location in the human body. The primary location of sensory neurons in the peripheral nervous system. These cells right here, I want you to look for them. These cells are found in the dorsal root ganglia attached to the spinal cord. So I'm going to show you these in just a minute. This second part of the image shows a somatic motor pathway. This is where we've been spending all our time. So this neuron is in the ventral horn of the spinal cord. And this is the nerve, the musculocutaneous, the radial, the sciatic, the tibial, nerves that you've learned. And you can see that these somatic motor neurons control skeletal muscles. This is where we've been spending all our time. This last one shows you a motor output, but this one is an autonomic motor. We will not discuss specific autonomic sensory pathways. There's a reason for that that I don't have to explain to you. You will understand later in your lives when you're going through autonomics. I require students in this class to be able to describe the basic anatomy of the motor outputs of both the somatic and autonomic nervous systems. This one I have finished. I will not go back and detail these. We'll go through a couple of slides in the next test section where I'll show them to you again. It's good to review. We'll just go through it a little bit, but I'm done with all the detailed teaching here. This part I'm not done with. So we're going to need to talk some more about this today, the autonomic nervous system. All right, so start getting the language straight in your mind now. Notice in my connection for autonomics, I have central and peripheral components. And you can see here that this neuron cell body is in the spinal cord. And the neuron cell body has an axon that goes out and talks to a second cell. And then this cell goes out and talks to my autonomic effector organs. I said it a moment ago. Cardiac muscle, smooth muscle, and glands. Now, these cell bodies, this shows a single cell body. But this cell body right here is going to become the centerpiece of the terminology that I'm going to teach you for autonomic pathways. It will become the centerpiece. And I'm going to use the word ganglion over and over and over. So I need to tell you what that is. Although you've heard this on occasion in other places, we've sort of briefly touched on it here and there. I'm going to really use it a lot today, so I want to use the word with you now and tell you what I mean by this. Okay? So we're going to have, in our outline here, the first piece you have is the nervous system can be divided into central and peripheral, and I will do that anatomically. The nervous system can then be divided into functional components, and we call those somatic and autonomic. Now, for appearance. So I can get the word ganglion in your heads for appearance. If you look at nervous system tissue, so this is a picture of the spinal cord here. If you look at nervous system tissue, anyone can clearly see that for the brain and the spinal cord, some of the tissue looks dark and some of the tissue looks light. Now, the reason for the appearance of tissue in the nervous system is primarily the presence or absence of a substance called myelin. A substance called myelin. Now, let me skip ahead for just a minute here and show you this. Obviously, we're going to do this again, okay? But this picture shows you a neuron cell body here, and you can see some sort of substance that is covering. You can see it here. Here's the axon of this cell. You can see this substance is actually covering the axon. The axon should look like that, right? It should have that kind of appearance to it, but it's covered with this stuff. This is called myelin, and there are cells, as we will show you, in the central and peripheral nervous system that do this. They cover the axons. Now you can also see that this myelin being produced here does not cover the entire axon. Instead, there are gaps between the pieces of myelin, and we call those gaps nodes. The axonal nodes are the nodes of Ranvier, R-A-N-V-I-E-R. I'm going to say that again. So the nodes are the gaps between the myelin pieces. Now, why is he making such a big deal in his structural description of the nervous system using myelin? Well, because when you see places in the nervous system that are gray, we call them gray matter, darker areas of the nervous system, you are looking primarily at areas where there are clusters of neuronal cell bodies that do not have myelin on them. And when you are looking at the nervous system and you see areas where there are white, lighter colored material, you are primarily looking at areas that are covered with myelin. So those are in fact the axons of the neuronal cell bodies. So if you look in the brain and you see gray matter, you think clusters of cell bodies. If you see white matter, you think these are bundles of axons that are going somewhere. Myelinated axons. Now, the last thing you'll learn about myelin from me after I show you the cell types in this presentation is that myelin is an important substance for nerve conduction velocities, how fast signals move. So, when an action potential runs down an axon, the way you learned it was the action potential just goes from one spot to the next. And that's the way it shows up on this presentation too. But that doesn't happen on myelinated axons. Instead, the action potentials occur at the bare spaces, at the gaps, at the nodes on the axons between the myelin. So the action potentials literally in our bodies on myelinated cells jump from one place to the next. And we're going to call that saltatory conduction. The word saltare means to jump. And so that's the way this works. It speeds things up greatly in our bodies. In fact, if you have a very well-known autoimmune disorder known as multiple sclerosis, this is the problem. The immune system attacks the myelin. And so over time, the disease causes the velocity of information in the nervous system to slow down. So people have trouble coordinating movement, walking, breathing, and other problems. MS is a major problem. It's a disease of this stuff. The immune system attacks the myelin. Okay, now finish up the vocab here. So if I say gray or if I say white, you know structurally what I'm talking about. If I'm talking about a cluster of cell bodies, unmyelinated gray matter, if I'm talking about this unmyelinated cluster of cell bodies, and I'm in the brain, I will call those areas nuclei. That's unfortunate, frankly. The neuroanatomy people and the basic biology people didn't get together on this. Because when I say nucleus to you, your first thought is a cell organelle that has the DNA in it, right? That's what a nucleus is. That's not what you mean when you say nucleus in the nervous system. A nucleus in the nervous system is a cluster of neuronal cell bodies. So for example, many years ago, as I was doing research in brain anatomy, I was interested in a brain structure called the suprachiasmatic nucleus. It is the cluster of neuronal cell bodies deep in your brain that is known to function as the biological clock. These neurons have the ability to tell time in the absence of timing cues. They are autonomous clocks residing in our brains. We call them the suprachiasmatic nucleus. Well, if I say suprachiasmatic nucleus and a student thinks that's a little place where the DNA is, then you've missed it, because this is a place where there's about 30,000 neurons all clustered together that have a singular function. So if I say nucleus, a person should think in the brain a cluster of neuronal cell bodies with a particular functional capacity. Okay? Gray matter, nuclei. If, on the other hand, I find a cluster of neuronal cell bodies in the peripheral nervous system associated with cranial nerves or associated with spinal nerves, I have a different name for them. I'm going to call those clusters ganglion, singular, or ganglia, plural. That will become the terminology that we will center on talking about the autonomic nervous system. The ganglia. Now, when I pointed to this guy a minute ago, I used the phrase dorsal root ganglion. That's going to come up again. A cluster of neuronal cell bodies. If I point to one of these cells right here, and I say it's part of the dorsal root ganglia of each of the 31 pairs of spinal nerves, you will remember from this slide that I'm talking about sensory information. So all these things will come together. This will be the second cell type I'll talk about, the unipolar neuron. They're found one place only, in the dorsal root ganglia of the spinal nerves. There are 31 pairs of these along the spinal cord, and there are no exceptions to this. Every bit of sensory information in the periphery, all of it, that passes to your spinal cord passes through unipolar cell bodies in clusters. Balls of cells that exist on the root attached to the spinal cord. We call them the dorsal root ganglia. Now, you'll know about them one day if you have a slipped disc in your back, because these ganglia sit in close proximity to the transverse processes. So if you injure your back and one of your fibrocartilage discs, the annulus, protrudes, sticks out a little bit, and presses this ganglia, this cluster of sensory cell bodies, up against the transverse process, you will know it. That will be interpreted as pain. And frankly, whether you interpret it as pain or it is pain is irrelevant. To you, it is pain. It feels like pain in your leg, right? But the pain is not in your leg. The pain is in the unipolar cell bodies being pushed up against the transverse processes of the bones. Ganglia. Ganglia will be the language I will use here, because in the autonomic nervous system, all of the descriptions will be a two-step process. The anatomy will be two steps. The first step will be here. We'll call this neuron the preganglionic cell, and we'll call this one the postganglionic cell. So this will be the language of the autonomic nervous system. These neuron cell bodies here, there will be many of them in these balls, in these clusters, and I'll locate them for you. These we will call the ganglion cells of the autonomic nervous system. So we will speak about autonomics as preganglionic and postganglionic, and there will be a synapse between them. Okay, that's your introduction to nervous system stuff. Let's put some chemicals on it, because I want to spend most of my time talking about neurochemistry with you. I will return to that nervous system stuff in test 4. but I want to get it all out in the open. I'm going to do a little bit more autonomic anatomy with you and physiology with you in here, and we'll do a lot more of it in the next test section. So I do that on purpose. I want to teach it to you, and I want you to learn it, and then I want to teach it to you again, and I want you to learn it a second time so that you don't forget it. All right, so this picture is intended to take what we just learned about the anatomy and put some chemical messengers on it. All right, the first one you know. The top of this picture shows you the somatic nervous system, and here's a cell body. There's a myelinated axon, and that axon is going to skeletal muscle. And so you're going to call this pathway a somatic motor pathway. It includes cell bodies in the central nervous system and axons that are part of spinal nerves in the peripheral nervous system. All things that you knew. Now notice the chemical that is released is called acetylcholine, but you should be able to do better than that, right? You guys should be able to now say, that's a motor neuron. And one motor neuron and all the skeletal muscle cells it touches are called a motor unit. And the action potential forms after resting membrane potentials are established. And in this cell, the action potentials will bop down this axon and get to the end and cause the release of a neurotransmitter that we call acetylcholine. And then the acetylcholine will bind to the sarcolemma and cause the sarcolemma to reach threshold. And then action potentials will occur along the sarcolemma, passing down the transverse tubules, causing calcium release from the terminal cisternae. Am I ringing bells? This is what you've just learned, the somatic pathway. This shows you the acetylcholine being released here. All right? The rest of this is the autonomic nervous system. And look at the chemistry here. First thing I want you to notice, both for sympathetic and parasympathetic, it will become the language of the ganglion. So this piece will be pre-ganglionic. This piece will be post-ganglionic. So in the sympathetic nervous system, the neurotransmitter released pre-ganglionic is acetylcholine. Isn't that interesting? The neurotransmitter released post-ganglionic onto smooth muscle, cardiac muscle, and glands is norepinephrine. In the parasympathetic nervous system, the preganglionic cell now releases acetylcholine. Isn't that interesting? And the postganglionic cell then also releases acetylcholine. Now, in the anatomy of these two, look at the anatomy. We'll just learn it in pieces, picking up pieces as we go. In the anatomy of this system, you can see that the preganglionic sympathetic axon is shorter than the preganglionic parasympathetic axon. Does everybody see that? That is a general rule, a general anatomical rule for these two systems. It does have exceptions, but this is a general rule. What this means is the ganglion cells in the sympathetic nervous system are close to the spinal cord. And the ganglion cells in the parasympathetic nervous system are far away. The axons of the preganglionic sympathetics are short. The axons of the preganglionic parasympathetics are long. Conversely, you can see the postganglionic sympathetic projections are long, whereas the postganglionic parasympathetic projections are short. So these are general rules. These are good rules to learn about the two systems. Sympathetic is short acetylcholine, long norepinephrine. The parasympathetic system is long acetylcholine, short acetylcholine. If you can remember that, it will take you a long way in describing the anatomy of the sympathetic and parasympathetic nervous system. Okay, so this is intended to really highlight the chemistry, the neurochemistry here in talking about neurotransmitters. All right, So, beat them to death with it. Don't let them leave it. Beat them to death with it. This is a typical pathway of a somatic. A typical pathway of a somatic motor output, showing this. The signal begins here in the brain in a structure called the precentral gyrus. Don't write that down. I don't want you writing that down yet. It's okay. I have mentioned it briefly in passing to you before, but I am not going to require that of you yet. Test 4, we're going to come back to it again, and I'm going to add some components to this. Now, some of you will remember, because I always show an image of the motor homunculus. Do y'all remember the motor homunculus? The map of the body on the precentral gyrus? Okay, so we'll come back to that again in the next chapter.